like to get started. We only have about half or a little under half of the class here, but that's all right, because what I wanted to start with is actually backing up and talking about Essay 3. So if you check in files again, in the files folder, you'll see that I've put, once again, a sort of outline, bullet points that give you some advice on how to handle Essay 3. And I also thought I'd walk through with you, hopefully getting some of you jumping in and giving me some answers. Not just Essay 3, but the lecture questions at the very end of the cardiac output section that we didn't really get to. And that's the questions 12 and 13. I know a few of you worked on it, but I'm going to reassign you guys today to new groups, so don't worry about that. I'm going to go through it with everybody, these last two lecture questions. So I actually wanted to start with 13 and go back to lecture question 12. And in doing that, you'll see 13 is really the heart of what's going on in Essay 3. It's kind of putting it all together. So I'm going to share my iPad with you, assuming all of this works. It's waiting. And kind of outlined here, some of the effects of exercise. Now, one of the things I would like to stress, and you'll see in that file that I gave you advice about, is if you start looking for similarities and differences between exercise and low blood volume, what you're going to see is that if you just tried to answer the question by what increases and what decreases, you're going to be in trouble, because you'll find they're both trying to increase cardiac output. They're both going to use the sympathetic nervous system to do that. They're going to do vasoconstriction. So you have to get more subtle than just what's increasing and what's decreasing. And you have to look at, for example, what is the starting point? What is the goal? What's the limit? How far up can it go? What will stop it from going any higher? And also, where exactly are these things happening and why? So I wanted to kind of lead you through some of this. So in exercise, the goal is going to be increasing your cardiac output above its resting level. So there is a limit to how much you can do that. And this is a term that is good if you have an Essay 3, but if it's not there, I would still just know it. And it's in the slides, and that's cardiac reserve, right? So at some point, if you are exercising intensely, you're going to get to the point where your heart is unable any longer to raise your cardiac output any higher than it's beating as strongly as the muscle of your heart is able to do. The heart rate is up high enough that perhaps venous return is actually getting lowered because of not enough fill time. And often people who do exercise call this hitting the wall, because you're trying to go harder intellectually, but your body is just not responding anymore. And because you're asking your muscles to work really hard and you're not able to deliver to them oxygen and nutrients, but mostly it's the oxygen, then the muscles start to not be able to perform even, as much as they were before, and you feel like, as I said, you hit the wall. So in the lecture slides, you'll see this defined, and you'll see that on average, what they give is you can raise your cardiac output about 5 times its normal level, and some people can, when they're really fit, can raise it up even higher. So let's take an average person here, and their cardiac output at about 5 liters per minute. So remember, that's what we calculated if we used a heart rate of 75 and a stroke volume of 70 mLs, we got about 5 liters per minute. And now they want to exercise and they want to raise their cardiac output. Maybe they want to triple it, so they want to have 15 liters per minute. So what are they going to have to do with their sympathetic nervous system? Let me just kind of randomly pick people and you can unsilence yourself. How about Lewis? What's going to happen to their parasympathetic? You might have to unmute yourself because I think I've got everybody muted. Okay, I'll pick somebody else. How about Michelle? What's your parasympathetic going to do?
It's going to go down, I believe.
Exactly. So you're going to have to silence this. Decrease your parasympathetic. Remember the parasympathetic, sympathetic are kind of the yin and the yang. They're going to do opposite things. If you decrease your parasympathetic, inherently that's all going to lead to a increased heart rate without even the sympathetic, right? That's going to lead to some increase in the heart rate because your pacemaker is now being released from its inhibition. And then your sympathetic, what's that going to do for the heart rate? How about, let's pick somebody else. Sarah, what's going to happen to your heart rate with the sympathetic? Increase or decrease is good enough.
Increase?
Yes, it's going to increase. And so that means that you're going to start a little higher in your pacemaker. The slope's going to be steeper. Your heart rate's going to increase. So these two things are kind of additive, right, on each other. They add to each other. You're going to decrease your parasympathetic. That's going to stimulate more of an increase in the heart rate just because the pacemaker is able to go faster. But now you're going to drive the pacemaker even faster. You're going to increase the heart rate. And in your cardiac contractile, what's the effect of the sympathetic in general? Let's pick somebody else. Maybe Alexis, what's the cardiac contractile going to be doing? It's going to use increase in calcium to do it, if that's a hint. Somebody else? Let me pick somebody else. How about Stephanie? What's the cardiac contractile going to do? Yes, increase its contractility. So it's going to increase contractility. It's going to contract stronger. So that term contractility means it's going to squeeze the blood harder. So that's going to lead to an increase in stroke volume, right? So with our formula that the cardiac output equals stroke volume times heart rate, both of those are increasing. So of course, cardiac output starts to rise. So the heart is working harder, but at this point, we haven't hit what is the major way in which we can increase cardiac output. What's the major influence according to the Frank-Starling law on cardiac output? How about Jeanette?
It tells us that this preload is a direct effect to increasing the stroke volume.
Exactly. So neither of these effects have really done the major thing you can do to help cardiac output. In order to do that, you want to increase venous return. And so that's where the shunting of the blood comes in. Now you have to put yourself into the shoes of some of your body as you are trying to exercise. Now let's assume you're exercising. What's going to generally happen is you're going to shunt blood from, you're going to try to shunt blood from almost all your arterioles, with the exception of your coronary vessels, because they have beta-2 receptors, your respiratory muscles, which also have beta-2 receptors, and your You're not going to change blood flow to your brain because they don't listen to this one way or the other. But most of the rest of the body is going to vasoconstrict. Well, you don't want to vasoconstrict your skeletal muscles. So today we're going to talk about this. Hopefully we'll get that far, that there are going to be local controls on those arterioles that are going to be considered intrinsic controls. And those intrinsic controls can override the extrinsic stimulus from the sympathetic. So the sympathetic and your skeletal muscles are going to say constrict, but your local controls are going to say, no, we need oxygen. And they're going to send out local signals that say, no, we're going to dilate here. So we're not going to constrict the skeletal muscles because of, so I'm going to make a list here, the skeletal muscles, and they're not going to constrict. They're going to dilate. Well, make sure not no sympathetic, but they're not going to be responding. And what about your GI tract? What's going to happen there? You think you'll constrict it? How about Selena? Dilate or constrict?
You're going to constrict since it's resting.
Mm-hmm. Yep, you're constricting your GI tract. In fact, most of your viscera, so I'm just going to add in the kidney. And that means you are going to increase venous return from that area. So I'm not going to go into lots of details, but let's pick one other area, the skin. So what happens when you're exercising? What do you look like? Do you look pale or do you look red and flushed? How about Angela?
You look red.
Right. That means you're not constricting, you're dilating. Now, why you're doing this is thermoregulation is overriding it. So it's not even the local tissues, it's your thermoregulatory negative feedback loop from your hypothalamus is saying, forget that, you're going to dilate here due to thermoregulation. So all of these things are going to increase your venous return. Now, the other thing you're going to be doing is you're going to be increasing venous return through your muscular pump. Because after all, you're moving. And what's going to happen to your respiratory system? How are you going to be breathing? Maybe Jennifer, how are you going to be breathing when you're exercising? More thoracic pump or less? Okay, how about Augustine? More or less thoracic pump?
Less.
When you're exercising, you're breathing faster. Yes, faster and deeper. So these two are also helping venous return and therefore preload. So all of this stuff here is increasing preload by increasing end diastolic volume through an increase in venous return. When you go to do this essay and you start looking at dehydration, then you are going to have some of the same things here. But for example, in dehydration, when we come to it, you're not going to vasoconstrict or vasodilate many places. You're going to do almost all vasoconstriction. So this part is going to contribute more than it did in exercise. On the other hand, when you're really dehydrated, you're not going to be moving around. So you may use your thoracic pump, but you're not going to use your muscular pump. So you're going to contribute less. So this is a subtle essay. You can't just say, up and down and this increases, that increases. You kind of have to say how much and from where and where are we going. So let's go to that one next. So in exercise, your goal was to get your cardiac output above resting level. And I think I did dehydration on another view. In fact, actually, no, I did do it on another note. Oh, I can go. Is it on this one? Yeah. Open. So shock is, and we're going to say hypovolemic here because this would be low blood volume. We're going to talk about on Thursday when we do blood pressure. But shock basically means low blood pressure. Lots of reasons that you might have low blood pressure. The one we're looking at is a low blood volume. So I'm not going to go through a minor dehydration like, I'm thirsty and I didn't get enough to drink. We're doing a dehydration significant enough that you have less water all over in your body. Since blood is largely made up of water, it's going to have a low blood volume. So this means hypovolemic, low blood volume. If you have a low blood volume, then what's going to happen to your cardiac output? Increase, decrease. How about Joan?
Decrease.
Joan?
Initial reaction is decreased.
Exactly. So because you have this low blood volume, this is going to decrease below normal, below resting. And our goal here is going to be to return it to resting. I'll put cardiac output to resting. So in both cases, exercise and in dehydration, you're trying to raise your cardiac output. But in one case, your limit is going to be your cardiac reserve, and you might be raising it 5 or more fold. If you're dehydration, then your goal is going to be to raise it up to normal, back to normal. So just like in exercise, you're going to decrease your parasympathetic and you're going to increase your sympathetic, and that's going to give you this rapid heart rate, right? So you decrease your parasympathetic and increase your sympathetic. So unless you're really pushing yourself in exercise, that rapid heart rate is helping with your cardiac output. Now, if somebody has very severe low blood volume and is in deep shock, their heart rate, their sympathetic might be going nuts, and their heart rate might actually be detrimental to returning the cardiac output to normal because you're not allowing the heart enough time to fill again. Let's just say that heart rate is increased, and we'll just put, if high enough, it's actually decreases, leads to a decrease in venous return, which leads to even more of a decrease in cardiac output. So this person's in real trouble because one of the things that helped you in exercise was raising your heart rate. Unless you're really pushing yourself to the wall, it's helping your cardiac output. But if someone's in deep shock, that rapid heart rate may actually mean their heart isn't given enough time to fill with new blood, venous return is decreased, and their cardiac output goes down. So whether it's because of the high heart rate or it's because of low venous return due to low blood volume, they're going to have weak contractions of the heart. Weak contractions of the heart, and we'll say due to decreased venous return, which leads to decreased preload. And preload, of course, is the most important factor in determining cardiac output. So the heart's trying, the sympathetic is saying contract harder, but since preload is so much more important than adding more calcium on influencing cardiac output, the heart is contracting only weakly. It really needs that venous return to cause a strong heart contraction. So even though you're getting a lot of sympathetic, so I'm going to say despite sympathetic, you're going to have a decrease in cardiac output. The heart is really working hard in this person. It's beating fast. It's attempting to contract, but low venous return means you need, you're not going to get much stroke volume out, and you're going to get a low cardiac output. So you're going to use your sympathetic to try and increase your venous return. The fact that their skin is pale tells you what? How about, who didn't I pick? Let me go. How about Michelle? Oh, no, I picked you before. Dina, what's happening with your skin?
The skin is contracting, and the vascularity to the skin is contracting to shunt the blood more towards the vital organs and to the heart for venous return.
So your shunt to the heart. Your skin's a huge organ, so it has a lot of blood. We even called it a blood reservoir when we did the skin last time. So you really look pale. This person looks pale. Since you aren't putting much blood to your skin, and you're generally not providing blood to the skeletal muscles of your body, that's your furnace. Those are two huge organs in your body. The person's going to feel cold. So they might start shivering if they're cold, right? That's a reflex. You don't want that. So if someone's in shock, what you want to do is keep them warm so that they don't start shivering and lose more body heat in trying to warm up their body with their skeletal muscles. So they're going to feel cold. Their breathing is going to be rapid and deep. And so what are they trying to do with increasing the rate and depth? How about Jasmine?
Can you repeat that? Sorry.
What are they going to be doing by increasing the rate and depth of their breathing?
I'm assuming that they're going to try and get the blood flow to go around faster around the body.
So it's not having to do with the vasoconstriction, but it does have to do with what you're saying. You're trying to get the blood back to the heart, but you're doing it through your thoracic pump. So you're increasing your thoracic pump. And that's going to try to increase your venous return and therefore preload. But can you use your muscular pump? Well, no, because you're not supplying your skeletal muscles with enough oxygen in order to move enough. So the person, if they're really severely dehydrated and hypovolemic shock, they're going to be too weak to do that. So that means less blood to the vessels, less O2 to skeletal muscle, no muscular pump. Okay, so this person is really feels crappy. They might be lying down, and we'll talk today about, or maybe Thursday, about how that helps with return to venous return, because if you're lying down, then your blood doesn't have to fight gravity to return to the heart. You don't have to use your muscular pump so much. And you might know that if you were to treat somebody who is in shock, one of the things you might want to do is have them lie down and raise their legs so that you can actually have gravity help return blood to the heart instead of, going against gravity. And you're going to try and keep them warm. So all of this has tried to increase venous return, but you really haven't solved the problem, because typically in someone in severe shock, because your blood volume is still low, and we haven't talked about where you are vasoconstricting. So let me go down here and look in the low blood volume case. And we'll just list the same things, vasoconstriction via sympathetic. And we just said the skin is going to vasoconstrict, and the skeletal muscles are going to vasoconstrict. Now, these two did not vasoconstrict in exercise. They, if anything, vasodilated. So there's a difference between low blood volume person and the exercising person. But a similarity will be the GI tract and the kidney. Those are going to vasoconstrict. So all of these areas vasoconstrict. What about the respiratory tract and the coronary vessels? They're dilating still. So that's just via the sympathetic. Now today we're going to be talking about the regulation of blood pressure. And lecture question 2 asks about renin-angiotensin. So in the case of this person who has low blood volume, you're also going to activate cells in the kidney called granular cells. So two things are going to happen in the kidney. This is smooth muscle, right? The smooth muscle to those blood vessels are going to constrict. So less blood's going to go to the kidney. If less blood goes to the kidney, that means you make less urine. If you make less urine, you lose less blood volume. And so that does help you make your blood volume try to return to normal. But it's pretty minor. And when we talk about the kidney and even on blood vessels later today, you'll see why it isn't the major. The major way is your kidney's granular cells are receiving both sympathetic and another stimulus, the low blood pressure. Those two stimuli together cause a large release of renin. Well, what happens in the exercise person? Well, the exercise person, if I change the color here, let's go say this is the exercise. In exercise, what's happening is you are increasing this, but then you've got high blood pressure. These cells listen to, these granular cells are trying to make a compromise between these two stimuli that are in opposition to each other. High blood pressure will tell the granular cells, if you're releasing renin, stop. Sympathetic saying, release some renin. So in an exercising person, it's not clear any renin would be released at all. Does that make sense? Let me let you think about that for a second. Any questions? Okay, so what about in the dehydrating person? If we go forward, back to black. That large renin release, that leads to angiotensin 2. And yeah, you should know the cascade of reactions, angiotensin 1 to angiotensin 2. We'll review that later if you've forgotten from 103A. And the targets of angiotensin 2, and this is kind of a medium-term solution, are going to reinforce vasoconstriction. And in this case, it's going to be pretty global. It's not going to vasoconstrict to your brain, your coronary vessels, your respiratory system. So I'll just say everywhere except coronary, your brain, and respiratory system. So now sympathetic is already stimulated that, but now angiotensin 2 is also stimulating it. So these are synergistic effects. If for some reason you're in low blood, running away from the tiger is what I used last year, and you're bleeding, you have low blood volume, you're still trying to run, the muscle might be saying, I'm trying to override the sympathetic here. But once you add the renin and the angiotensin 2 to that, it's going to be very difficult for that person, skeletal muscles, to override and say locally, I need this oxygen, and you're going to end up vasoconstricting your skeletal muscles. So this is sort of a medium timing wise. The fast thing is your sympathetic response because it's neural, and then you get this renin angiotensin vasoconstriction that kind of happens pretty fast, a little slower than just pure sympathetic. And then you have the real solution here, which I'm going to change to red since it's blood. And what I mean by real solution is you're going to increase aldosterone, and you're going to increase ADH. And in fact, oops, that should be an A. Can't talk and write. ADH. And in fact, if your blood volume is low enough, that low blood volume will directly stimulate ADH. So you'll have 2 stimuli for ADH. So if you've forgotten, this causes an increase in water reabsorbed, but water alone cannot move without a salt to follow. And when we do the kidney later, you'll see that some of that's inherent in the kidney, but you're going to supplement that by an increased sodium reabsorption. And together, what this is going to do is increase your blood volume. So we don't see that in the exercising person because they aren't releasing much renin, if any at all, unless they become dehydrated by exercising, which don't worry about that in your essay. Don't push that exercising person to the point of dehydration. Just say it's an initially exercising person, they're raising their cardiac output. Their heart is working harder when they're exercising because it's contracting harder. Now, in the dehydrated person, their heart is working harder, and for exercise, that's going to be hopefully a short term, unless you're even if you're running a marathon, it's going to be a matter of hours. But if your blood volume is low and your heart is working harder and you're still low cardiac output, that means that all that time your heart is working harder, your organs are not being served enough oxygen. And that person, if they're dehydrated enough, eventually the organs are going to not get enough oxygen to survive. And people with severe hypovolemic shock die of organ failure because they're not getting enough blood. Well, why are they not getting enough blood? Because we're vasoconstricting to try and raise cardiac output, since the only way we can get the heart to work harder in these initial stages is to make it, the only way we get cardiac output up in initial stages is to make the heart work harder. The only long-term solution to that is to increase our blood volume back towards normal, and that's going to let the sympathetic go down, less renin will be released as blood pressure returns to normal. So when you think about it, if someone's in shock and an EMT arrives, one of the first things they're told to do is to give the person saline, to put a needle in their vein and give them saline. They're not giving them blood, whole blood. I mean, they might if they were in the hospital, because then they could do all the tests necessary to know that they're giving them the right blood type, et cetera. But not in the EMT, not out in the field. What they're doing is just giving them saline solution. Now, why they're doing that is they're trying to raise their blood volume. They're raising it by adding not water back, even though this person is, say, dehydrated or low blood volume. They're trying to just raise the volume overall. when you raise that volume of blood up, preload goes up, venous return goes up, and diastolic volume goes up, and you can raise the cardiac output. Does this all make sense? Is there any, good. All right, so remember in the case of the dehydrated person, you're really using your kidney and renin-angiotensin to address the final problem. And of course, if you've done this, urine output is going to fall. So you're going to be making less urine too. That's another effect that you'd see in a low blood volume case that you won't see in the exercise case. Or it's minor. Let me see if I've got a chat here. Is that? Oh, I see. Jennifer answered by chatting and I didn't have it open. Sorry. Thank you for answering. Okay. All right. So let's go to our groups again. And I posted, let me escape this share. And I did post the questions. So let me share again the questions and break you guys into groups. So if you've got this, this is in announcements, and I broke them, as you can see, into groups A. A has 3 related questions. The why some have 3, some have 2, or even 1. Depends on how long the answers are. But I also tried to break this up into questions that are kind of grouped around a common topic. So the topic we're addressing today is hemodynamics. And what I mean by that, and the book means by that, are what are the factors that affect blood flow through the veins. So up until just a moment ago, we were really focused on the heart. And when we talked about blood vessels, it was to serve more blood back to the heart. We vasoconstricted them, diverted blood back to the heart or not. Now we're going to look at the other side at how does the heart or the vessels affect the heart's ability to get blood into them. So that if it's harder to get blood to go into a blood vessel, that blood vessel has greater resistance to flow. So the first group is going to be talking about laminar flow. If you could please define or describe what laminar flow is as best as you can. And the most important factor that affects resistance is diameter. So we saw how vasoconstriction changed the diameter and therefore changed the ability of blood to enter into, say, a capillary bed or be diverted back to the heart. And then the other 2 factors are touched on here in Group A. Now, Group B is really looking at some of the things we'll be doing in lab on Thursday when we look at blood pressure. So talk about what these things are and explain the variables that help describe how the vessels affect cardiac output. When you do this, there is a formula. It's not a formula you're going to plug numbers into, but can you please go over that formula for how resistance and blood pressure affect cardiac output? And then, then the next group is going to be looking at something we've already covered. So this first part is pretty easy. What are the primary sources of venous pressure? And then the negative feedback loop that helps maintain blood pressure using short-term mechanisms. So when you do the negative feedback loop, you should have who are the receptors, what are they detecting, what are they doing, who are they talking to, where's the control center, et cetera. And then the next group, D, looks at, well, what else is this control center listening to? What other signals are talking to? And then what hormones are important? So these 2 are a bit, we just covered this part. They're not, these 2 aren't directly related to each other, but I kind of lumped them together. And this one should be pretty easy because we just kind of covered it. And then the next are a new topic, and that's looking at some factors that affect, well, this one isn't really new. This one is kind of stuff we just covered, but putting it into an anatomical context, short-term and long-term responses. That's what that's standing for. So these two are a bit unrelated as well. And this is a new topic. What are these intrinsic factors? So we're going to spend some time talking about that because I think in the book it ends up being kind of list-like. So if you can try and put them into some context. Now, one of those factors is this one, and I put that guy in another group because this one, well, I just put him over there because otherwise this last one would have too little to talk about. And then hopefully we'll get to 18. We'll see. So in going over that, you've hopefully heard all of them. Let me now put you guys into groups. And I'm going to try and mix it up in a little bit. Let's see, I believe I've got, what I have, A, B, C, D, E, F, right? So that means we need 6 rooms. And I'm going to just try and mix you up a little so you're not always with the same people. So let's create the breakout rooms. So maybe we'll go every 4th person or something. Okay, 1, 2, 3, 4. We'll put 4 people in this. Well, maybe I'll give 5. Okay, it looks like we're back. So I'm going to silence someone. Let's see if we can do it this way. You don't. Okay, so I'm muting everybody because there's some background noise. But remember, you are going to have a spokesperson, and if we want to get through this material, that person needs to volunteer. Last time I did the list by just alphabetical, so I had an idea who was in each group. This time I don't, since I kind of randomly threw you in. So if you could please jump up and, as your spokesperson, start to explain your question. So for Group A, the probably the hardest concept in Group A is this idea of laminar flow. So can the spokesperson from Group A explain what laminar flow is?
Yes, that would be me. So laminar flow describes the different levels of resistance in blood flow through the blood vessels. So the closer the blood closest to the vessel walls encounters more resistance than the blood flowing more through like the exact center.
And that's because of what's happening to that blood that's touching the wall.
It's encountering friction.
Right. So there's more friction along the walls and less friction in the middle. So the way they symbolize that was the smaller, shorter arrow, right? Okay. So, and then this is going faster. Good. That's laminar flow, the idea that you have more friction along the walls and less friction in the middle. So what would happen with Diane? Who was speaking? Was that Ileana?
Yeah.
Yeah. So what happens if I make this diameter smaller then?
So there's less room in the middle for that blood with the decreased friction to go through. So more of
Exactly.
it.
Yes. So if, let's say here we had 70% of the blood going through the middle and 30% going around the edges, now suddenly we have, 30% going in the middle. And so we'll put this as 30% in the middle, and we have 70% going around the edges. So Ileana, there was a formula, which you don't have to put any numbers in, that express this. Can you give me that?
We didn't discuss the formula.
So resistance is proportional to the diameter raised to, or the radius raised to the fourth power, right?
Oh, yes, radius
Remember that one?
to the fourth power, yes.
Yeah, so please include this in your discussion, because when you raise something to a power, that means, this is going up like exponentially. I mean, this isn't raised to the 10 power, but if you have the diameter, then you're going to take that 2 times 2 times 2 times 2 times 2. You're increasing the resistance substantially. Okay, so make sure that you include that idea, because diameter is the most significant factor that they're going to cover. Kind of like preload was so important, it's the most significant to resistance in determining resistance. Okay, so was there anything else in that, Ileana, as far as, the next question is, what about taller people?
So a taller person would have longer vessels, and resistance is also proportional to the length of the vessels.
So resistance is proportional to length. And that kind of just makes sense, because if you're having blood rub along a wall of a vessel, doesn't matter the diameter, the longer that vessel is before it gets back to the heart, the more chance it has to rub, and therefore resistance goes up. So in general, taller people have greater resistance. Their cardiac output needs to be the same. Their tissues are asking for the same amount of blood as a shorter person. So what you'll find is taller people generally have higher blood pressure. And we'll get to that in a minute when we do that formula for cardiac output. And then what's the last piece, Ileana?
The blood doping one?
Mm-hmm.
So blood doping increases the viscosity of the blood because there's more red blood cells in it. So it's more viscous, it's thicker. So there's increased resistance, which leads to higher.
So some people might not know what blood doping is. What do you mean by blood doping?
Blood doping is when you inject more. What says on the paper? Injecting packed red. I can't talk.
That's okay. I'm fine.
Red blood cells into the bloodstream. So I'm not 100% sure how to explain it, but I guess.
So why you do that is athletes do this as a way of cheating. So one way of blood doping is what Ileana said, which was they, well before the competition, months before, they have blood drawn, and then they centrifuge it, so they separate the watery portion from, or most of the watery portion from the red blood cells. And they then have, right before the competition, they take that blood out and they put it into just the cells, not the plasma, the fluid portion, back into their blood. So they have a higher number of red blood cells than they normally would, meaning it can carry more oxygen. But it also means resistance to blood flow goes up. So you can think of this, an analogy I used in the lecture was like using mud flowing through something versus water flowing through. You're going to have more friction when there are more things to rub, so a denser red blood cell. So when we talked about dehydration is kind of not blood doping, but it's another way to increase viscosity. If somebody is severely dehydrated, then the fluid part of their blood, their plasma is lower, and therefore their red blood cells are higher. The same thing happens with people who take steroids in order to increase their their red blood cell production and try to cheat that way. Again, the viscosity goes up. All right, great. Thank you, guys. So those are the factors that are influencing resistance to blood flow. So as the blood flows from, let me go down here, from the large arteries to the venules, and this is SA4. It even refers to this figure. So make sure that you can understand this both graphically and conceptually. So we're going to have a systolic pressure of 120. Oops. I don't know why it's not doing that. It's on the pencil. Okay, it's taking a delay, but 120 millimeters of mercury. So when you're leaving the heart, at the moment when it's contracting the hardest, the blood pressure that's leaving is 120. And it goes through the aorta. So let's put it as, I don't know why this is being so slow. The aorta. Guess it's trying to talk or something. Oh, God. Got to be some craziness going on. I think I have to do that, don't I? Maybe I just do that. But get rid of it that way. That's what you do. Okay. So the aorta, and then it goes to the large vessels, large arteries, and then it goes to the smaller arterioles. Dang. I keep hitting something. Sorry. Maybe that makes it go away. Small arterioles. And then it gets into the capillaries and the venules and the veins. Maybe because I'm trying to write this when it's transmitting it over to you guys. So you're going to stay with something called a mean arterial pressure over here. We'll talk about what mean arterial pressure is in a second. It's going to stay pretty flat as it goes from between 120 and 80. I didn't do that very well. Let me erase that. Erase this too. So we need a number in between there. Put 80 here. It's going to be going between 120 and 80 as the heart beats and relaxes, diastole, systole. And then it's going to get into these large arteries. Oops, it's still going to stay somewhere around 120 to 80. You're watching the heart beat here as it goes up and down. And as the arteries branch and get more of them, you start to see a decrease. As it goes into the arterioles, it decreases quite a bit. Then it gets to the capillaries, it decreases. And by the time it's in the venules, and you don't really see the systole, that's probably not very well done. In the veins, it's almost down to zero. So the next group, who is their spokesperson? Who's the spokesperson for B? Somebody speak up. I'm going to use this graph while you explain these terms.
I will do it.
Okay. And who is this? Is this Alex?
Yeah.
Okay, Alex. So the first thing says compare and contrast pulse pressure and mean arterial pressure versus systolic pressure. So can you just combine those or whatever?
Yeah, so pulse pressure is a difference between systolic and diastolic. So the systolic is 120 and the diastolic is 40. So the pulse pressure will be, diastolic is 80. So the pulse pressure will be 40.
Right. So when you take your pulse and you're feeling a little bump on your radial vein, say, or your carotid artery, radial artery or carotid artery, what you're feeling is a difference, let me change the color, between this value and this value. So it never goes down to zero, and we'll talk about what diastolic pressure is, but that difference here is pulse pressure. So that's the difference between diastolic pressure, when the heart is relaxed and the ventricles are relaxed, and systolic pressure when they are contracting. Okay, and what about mean arterial pressure?
It's a pressure in an average blood. Mean arterial pressure is an average blood pressure in an individual during a single cycle.
Okay, so he's right. You have to have time involved here. That time is that single cycle. So how much time does it take to go from systole to diastole? And then what's the average pressure in that time? Now, why that's important is that that's really the pressure head overall that the pipes are seeing. Yes, they get a pulse of 120 and then it drops down to 80. But on average, we want to know what is the pressure that's pushing blood from point A to point B, from the aorta to the large arteries, to the small arteries, to the capillaries, etc. So what is the average pressure behind this? Now, because the heart spends 2/3 of the time of its cycle, one cycle, in diastole, this 80 becomes 2/3 of the mean arterial pressure value. And it only spends 1/3 of its time in systole. So when we take an average over a cycle, we can't just say, well, what's the average of 120 over 80 evenly? We can't just add those together. That would be 200, right? And divide by 2 and say it's 100. It's not. It's biased towards diastole. So in lab on Thursday, we'll work more with the formulas. But in general, if you have this blood pressure 120 over 80, your mean arterial pressure, if it will write it, is generally around, and you'll have to know this number, around 93. So when it's leaving the heart, it's about 93. As it goes down, and this is what the graph looks like, you see this big drop in mean arterial pressure until you get to around the capillaries. So in the large arterioles, it might be, in the 80s. And then you see a sharp drop in the small arterioles. So when you do this essay, tell me why that drop occurs from what Ileana was just explaining. What happens when you think of the diameter and the length? Viscosity we won't worry about. What happens to the vessels as they start branching out? What happens to their length? It gets longer. What happens to their diameter? It starts getting smaller and smaller. So this drop is due to resistance increase. I know it's taking forever. Oh, well, we'll just let it slowly take forever, increase with increasing length. Now it's going to go fast. And decreasing diameter of these vessels. Okay, now it says you need to know this graph quantitatively. So the numbers I'm putting on this, the y-axis, you're going to need to know these, because when it gets to the capillaries, this is a key number we're going to use later, it's around 35 at the entrance to the capillaries right here. When it leaves the capillaries over here, it's at 17. And we are going to use those numbers. Eventually, when it gets to the large veins like the vena cava, it's pretty close to 0. And that's why you need your thoracic and muscular pump. Okay, so the last thing that you guys had to do was the formula, right?
Yeah, it's diastolic pressure plus pulse pressure divided by 3.
So for mean arterial pressure, that equals 2/3 diastolic plus 1/3 systolic is what I explained. Now, what he just gave is the formula that they give in the book, which is going to come out to, if you do the math, it'll come out exactly the same. So it's diastolic pressure plus pulse pressure divided by 3. So if you do the math, you'll see both of these come out to 93 if you're using 80 and 120 as your average systolic and diastolic pressures. Now that is, that's not the actual formula that I was looking for. What I was looking for, and anybody in the group, because it sounds like you misunderstood what I was looking for. It's the formula that cardiac output is equal to delta P over R. So remember, cardiac output is also equal to stroke volume times heart rate, right? So this side is the heart's contribution, and this side is the vessel's influence. And again, you don't have to put numbers onto this side of the formula, because here you do have to know numbers, because resistance is then inversely proportional to cardiac output. If resistance goes up, like when Ileana was talking about a taller person, when this number goes up, cardiac output, the need of the body stays the same, and you're trying to maintain that resting level of cardiac output. So what is your only alternative? It's to change the pressure. The only way you can change the pressure is to make the heart beat harder so that your mean arterial pressure increases to overcome this increasing resistance. So another way besides being tall to have longer vessels is to get fat. So obese people tend to have high systolic and diastolic blood pressure, or at least high systolic pressure. Why do they have higher pressure? Because they're trying to increase this. So if this increases the resistance because you got fatter and longer, then this has to increase as well. Does that make sense?
Yeah.
Okay. So we finished, I think, essay 4 now, if I've got my essays floating around here somewhere. We finished, here it is, essay 4. For the first half of it, I guess we haven't finished the second half, which is where we're going next. Okay, who is Group C's spokesperson?
That's me.
Okay, Kaylin.
No, Dina.
Oh, Dina. I saw somebody else's picture pop up, so. Okay, Dina, so tell me about short-term negative feedback of blood pressure.
I've got 2 primary sources of venous pressure. It's number 11. We're on question 11, right?
Oh, right. I'm sorry. Yes, that was the easy part, right?
Okay. So the 2 primary sources of venous pressure that brings blood back to the heart, and the force that aids in return from the vessels of the head and neck. The pumps in the form of the muscular and thoracic pumps create the pressure that returns blood to the heart. Skeletal muscle, particularly in the appendages, function as muscular pumps that contract to squeeze or compress the veins and assist in pushing blood back toward the heart. The thoracic pump functions on inspiration to decrease pressure in the thoracic cavity while increasing pressure in the abdomen. Pressure in the abdomen results in further assistance via venous compression or squeezing, while the decreased pressure in the thoracic cavity relaxes around the veins, decreasing resistance and kind of sucking the blood flow back into the right atrium. Gravity is the third force, and that aids in venous return from the head and the neck.
Brilliant. Okay, that's perfect. Okay, so now let's jump to the short-term negative feedback.
Right. So the negative feedback loop that uses short-term mechanisms, blah, blah, blah. The neural controls provide a negative feedback loop to provide short-term maintenance of cardiac output. The baroreceptors in the aortic arch and carotid sinuses respond to the stretch from arterial pressure. When blood pressure decreases, baroreceptors are inhibited, which decreases impulses from the baroreceptors to the medulla oblongata. This decreased activity activates the cardioacceleratory center and stimulates the vasomotor center while inhibiting the cardioinhibitory center. Increased sympathetic impulses to the heart in the form of norepinephrine, epinephrine, increase contractility and vessel constriction to increase venous return and improve preload to increase efficiency of stroke volume and increase cardiac output and return blood pressure to normal ranges. On the other side, in response to increased blood pressure, the baroreceptors are stimulated by the increased stretch and pressure to the aortic arch and carotid sinuses. This in turn sends impulses to the medulla oblongata to stimulate the cardioinhibitory center and inhibits both cardioacceleratory center and the vasomotor center. Decreased sympathetic impulses reduce heart rate, contractility, and cardiac output, while decreased rate of vasomotor impulses allows vasodilation to occur and reduces total peripheral resistance. Decreased cardiac output and resistance returns blood pressure to the heart.
Okay, that's great. So,
Helpful.
before class, and since this is being so slow to respond, I think this is it. Let me just take a look. I thought I'd write this up, what she just went through. So, first of all, as Dina mentioned, in your medulla, you have a cardiovascular center. This is one of our negative feedback control centers, and it's listening to multiple receptors. So the next group is going to say some of the other receptors it's listening to. But the one receptor that we're focused on now are the stretch receptors in the carotid and aortic bodies. Now, these bodies are just like stretch receptors in a way in your skeletal muscle. These are modified smooth muscle fibers that are embedded in the carotid tunica media. And like stretch receptors that you learned about in skeletal muscles, they are mechanoreceptors. And being a sensory, they adapt. If you remember, and I underline that as an important point, because this adaptation means that if you are counting on these baroreceptors, they are really good when you have you want a fast response to blood pressure to change your mean arterial pressure, your systolic pressure. And but if you have some sort of chronic change in it, so that you are chronically having an elevated blood pressure, like someone who's obese, they just adapt to that new value. So it's not like an obese person's carotid and aortic baroreceptors are chronically trying to lower their blood pressure. Unfortunately, no, they've just reset to a higher set point. That becomes the new normal. And so, yeah, they're still going to use their negative feedback loop, but they're going to bring it to a higher blood pressure because, of course, they need that to serve all those longer vessels. So here it is written out, and you saw it on figures in the slides that you studied beforehand. You have a higher systolic pressure, that's the variable. These guys are the receptors for this short-term response, and they're in the aortic and the carotid. They decrease when there's when there's decreased stretch because blood pressure drops, then you see decrease in action potentials from those baroreceptors to the cardiovascular nuclei. Now, just like Dina went through, that means you're going to stimulate sympathetic. You're also going to inhibit the cardioinhibitory nuclei. So there are two nuclei, the cardioacceleratory and the cardioinhibitory. Great names because easy to remember. Once you activate sympathetic, you are going to cause greater contractility of the heart. It's going to contract harder. And you're going to also cause vasoconstriction over here, shunting blood back, which will increase preload, just like Dina mentioned. And you are going to stimulate renin in this person, and that's going to help you vasoconstrict too. You're going to increase preload. Preload is that's why I made this arrow fatter, the strongest, most important way to increase your contractility, how hard you're contracting the heart, and therefore increase stroke volume and increase mean arterial pressure, blood pressure that's coming out. All this makes sense? Okay, so she did this as well. You're basically reversing this. You're going to use the cardioinhibitory. So if you wanted to, let's see if it'll respond fast enough now. If you wanted to reverse this, then if you had blood pressure is greater than the set point, I don't know why it's writing fast now, but it is. Okay, then you want to, you have a higher pressure here. You're going to get greater stretch than you expect. Therefore, you're going to increase the action potentials. That's going to increase your parasympathetic, decrease your sympathetic. You're going to have less contractility. You're not going to get any renin. And you're going to dilate these guys, so this is all going to drop out, and therefore you'll lower your blood pressure. So one of the key points that I don't think is even mentioned in the book is this idea that these guys adapt, but they are a sensory receptor, and they will reset the set point if they're chronically stimulated. Okay, so how much time have we got? Oh, we only have 5 minutes. So let's go to the next group and say, who else talks to the control center here? Who's the spokesperson for the next group?
I'm the spokesperson for that group.
And who is this?
Jeanette, it's Jeanette.
Okay. All right, Jeanette, so who else talks to this cardiovascular nucleus?
So the other factor are, instead of the baroreceptors, we have chemoreceptors, which respond to responses in blood pH, O2, and CO2. And this would because if you have the increase in body temperature or trauma to the body, the chemoreceptors would activate or deactivate the vasomotor center or inhibit or stimulate the cardiac acceleration center.
And where are these chemoreceptors?
In your blood vessels.
In the aortic and carotid body. There are two receptors in the same body. Maybe that's why they're called bodies. I don't know that they're the same cells. And as she mentioned, they are looking at your blood pH and CO2, largely because you're going to regulate your respiratory system. You want to synchronize the respiratory system. So one of the outputs of the cardiovascular nuclei and the respiratory nuclei is they're going to talk to each other because, of course, it makes no sense to increase your heart rate and depth if you're not increasing your respiratory rate and depth. That way you will be bringing more oxygen into your body so that the blood, as it rushes into the lungs, can get more oxygen. So you want to synchronize those systems. Are there any other inputs that you can think of? What other times does your heart rate go up and your that don't have to do with your blood pressure being abnormal? Let's say you take an exam. what happens to your cardiovascular center?
I think the other inputs would be the limbic system and emotions, as well as like proprioceptors in the skeletal muscle when you're exercising and increasing blood pressure.
Great. So you actually get a little reinforcement from the stretch receptors in your skeletal muscles. So when you activate it during exercise, that will feed back and kind of reinforce, yes, we need more sympathetic, we need more contractility. And then limbic, all the E divisions. So these are all E division things for your emotions. You could be just, upset, and you don't really need a rise in your blood pressure, but you do get it because there are other inputs on this cardiovascular nucleus. Okay, in the last couple minutes, Jeanette, what were the hormones that act to increase cardiac output? This is a review from where we were.
It would be the antidiuretic hormone and the aldosterone from the renin-angiotensin mechanism.
Exactly. Now, there is a hormone that opposes this, and that's from 103A. Let me just put it down here. So we just said the hormones were epinephrine, sort of norepinephrine too, and angio-2. Another one, these are all going to increase blood pressure. Another one is ADH. ADH is sometimes called vasopressin because it causes constriction of the blood vessels. How about if we want to decrease? This is when your heart is saying, "Uncle, I can't do it anymore," and the heart itself releases a hormone. Anyone remember that hormone? I know it was a long
Was it prostacyclin? Prostacyclin?
Oh, that's a great guess, but no, it's atrial natriuretic peptide. Do you remember ANP? If not, go back and review that. There's actually a really good picture for ANP later in the blood pressure chapter, so we will revisit it. But ANP basically blocks aldosterone, blocks ADH, blocks angiotensin II's effects, so that you lower your lower the blood volume, therefore lower the blood pressure, and it's a vasodilator. And it's only released when blood pressure is, if you want to, I'm sorry, this should say, to be clear, want to do these things. Oops, I can't write want, huh? Want to. This is the goal. You want to increase your blood pressure. Here you want to decrease your blood pressure. So this opposes aldosterone, ADH, and angiotensin II. So it doesn't oppose the sympathetic, but it does lower your blood pressure by lowering your blood volume. Okay, so we got up to 14. We have 4 more questions, and one of them goes into the long-term response. Now that's going back to the kidney and looking at long-term ways in which blood pressure is and we did this. So I'm just going to, I know we're a little bit over, but just quickly point out that we did follow this, but what I really wanted already, we talked about the granular cells, renin, and all the effects of it. But what I really wanted to stress is that these guys are not going to adapt. So when you have a chronic problem with your blood pressure, your kidneys, if it's low blood pressure, are going to continue to release renin. Now, if it's high blood pressure, they are just not going to be releasing renin when you may need it, but they don't really help with the high blood pressure that's chronic. So their response is only, unlike the baroreceptors, which can cause higher rate of action potential, lower rate of action potential, constantly monitoring blood pressure. These guys, their only output is to increase blood pressure through renin-angiotensin. So if your blood pressure is chronically high, you're going to, if anything, just suppress renin but renin's only released if you have low blood pressure problems, right? So, yeah, so they don't help you a lot with chronic high blood pressure. If, on the other hand, you have low blood pressure, we did that with the dehydration, low blood volume person, then they do help. So I think you've got all of essay 4. When we come back on Thursday, we'll finish this up looking at how those numbers back, did I still have it in this one? No, it's in a different one. On the graph where we did mean arterial pressure and systolic pressure and pulse pressure, how those then allow for capillary exchange when you perfuse the tissues. So that's the last topic. We'll also start into blood. So we're going to jump over to blood. So for lab today, I'm going to do an introduction of them, but you're going to spend the entire time really looking at pictures of models and your pictures in your book. And I'll introduce it at the beginning. So I'll see you guys in 10 minutes or so if you're in the first lab or want to be in the first lab, and give you a break to go get something to drink or eat or pee or whatever you need to do. Okay, so any questions before I leave?
Dr. Kate, can you explain the adaptiveness again for the baroreceptors versus the long-term regulation adaptive?
So if you go back to the sensory chapter, you can read about this phenomenon called adaptation. Probably the place that you might relate to it easiest is if you are wearing clothes, which hopefully most of you are. If you're wearing clothes, after a while, you don't feel the clothes on your body anymore. And that's partly because your brain starts to ignore things like that, but also the receptors, unless something changes, a sensory receptor is most interested in change, and therefore it doesn't send signals unless it's seeing a change in a stimulus, not just a stimulus. Maybe another way to relate to it is if you're living somewhere where there's an odor, after a while you don't notice it. So that's what adaptation is. It's actually at the end of the receptor end, not the central nervous system end, where it stops sending signals once that signal is kind of chronic and unchanging. That's what adaptation is. Okay, good question. Other questions? Okay, so I will see you in about 10 minutes. First, to show you what you need to work on. And this is going to go beyond the time you have in lab. There isn't any way to learn all this just during this lab time. If you would like, I will put you into breakout rooms. So let me know at the end if there's something you'd like to do by putting it in chat, and then I'll take groups and break them out so you can study together. So let me share this screen. Let me see if it's the one on the top. Nope, of course not. That has that's not it. It's a study guide. So I'm in files, and so I'm going to share that screen with you and walk through the things you'll be working on. Okay, so it's probably all pushed off to the side here, and I've got to get it over here. I did back up. This is our study guide, where your artery, vein, and nerve. Just to point out something, since I didn't show you these, that I put quite a few functions into each of these things you were labeling when you turned in your slides last time. So if I were to ask a function, you've got quite a list. We haven't touched on all of the functions that you see here yet, but we will talk about them more in lecture. Most of these we did talk about. So let me go past this. Again, this might be helpful for your lecture questions, because one of them asks about differences in arteries and veins. And And then we come to this slide that talks about dissection techniques. So one of the sad parts of doing this totally online is you don't get to actually do some of this stuff. If you were lucky enough in 103A that you got to dissect the cadaver, then you got a little experience, since we didn't leave until after the muscle project was finished. But I kind of walk people through some of these concepts when they first do a dissection. So what I'd like you to do with this is to study this information, so that if in the future you get a chance to do some dissection, or in the past, if they hadn't explained this, then you understand what these terms mean. So one of the words to the wise, this first one seems like it ought to be obvious, but you don't want to cut something until you know what it is. So you can't work blindly when you're doing dissection, or you'll ruin the animal or the cadaver that you're working on, because you'll cut something you don't intend to. So the other two terms, sharp and blunt, also kind of refer to attempting not to cut something unless you really need to cut it. So sharp dissection means using, the blade of the scalpel, or the edges of the cutting edges of scissors. And then you're actively, separating tissue by cutting through all the layers of a tissue. When you use blunt dissection, what you're often trying to do is pull apart the connective tissues that are holding things together, and isolate an organ or a structure by separating it from its connective tissue, and therefore separating it from the surrounding tissues. If you had a chance to do the fetal pig dissection, which is what you would do if we were in person, to look at their blood vessels, that's helped by having the blood vessels filled with latex. So that makes them sturdier. When you do blunt dissection, you're often using only slightly sharp things, like the points of the scissors, or the point of the scalpel, or the back side of the scalpel, or you might just be pulling things with forceps, grabbing on and pulling things apart. It's a bit safer, because although if you pull something delicate hard enough, you could rip it, if you're just gently pulling it apart, you're unlikely to damage anything you really want to look at. So you're just separating it, as it says down here, at the fascia. So study this slide for its concepts. So if I were to ask in the test, two things you should do before, one thing you should do before you do any dissection, and one is to identify it, what you're dissecting, and then be able to tell me sharp versus blunt, and what kinds of tools you might use to do either of those things. So given that we can't do a fetal pig or a cadaver, then in mastering A&P, again, I wouldn't suggest you do this during our lab time. You could do this outside of lab time. Then you can go in and study through the study area. So remember, you go into mastering A&P. It doesn't matter which of the two you go in, because I didn't make an assignment. You're just going to use the tools they have there. You're going to go on the left side, look for study area, and then open Practice Anatomy Lab. That's what PAL stands for. And it's got links there for you to look at dissections of a fetal pig. You're going to look at the cardiovascular system, and in particular, the dissection of blood vessels. So this is imitating what you would have done if we were meeting in person. There's things like quiz and stuff in there, and they may help, but if a blood vessel is not one on our study list for the human, then you don't need to identify it. If I remember rightly, they don't do any obscure, smaller vessels, that you wouldn't need to know in their quiz. In the human dissection, however, they do go beyond what we're doing. You're welcome to look at it, but even if we were meeting in person, I don't have you identify blood vessels except the ones that are attached right to the heart in the cadaver. So you don't use those for the test anyways. But if you're interested, they do have pictures of a cadaver dissection. Okay, then there is a handout called The Vessels to Know. It's in files. It's in this same folder. And it is your list. Now, this list, as I'll show you in a second, was generated from the lab manual photographs or drawings. And initially, I made the study guide using unlabeled pictures of the drawings in the lab manual. And then later I found this, it's part of the Mariab, and it's in the textbook, and I'll show it to you in a minute, because not only will you have to identify the vessels in these photo micrographs of the models largely, or drawings from the lab manual, that's what I'm mostly gonna do. There's a few I could ask in the pig, but the vast majority will be the photographs that are in the gross anatomy folder in under files in Canvas, or it could be pictures from the lab manual. So that's where that list comes from. The list is scarily long. But as I mentioned last time, the good news is well, first of all, almost all of the arteries have a vein with the same name, with exceptions around the heart. Most of them have the same name. The more challenging part is you do have veins where there's no analogous artery, and so some vein names are additional. And then in the other part that helps you is many of them are named after the landmarks. So if you know which is the radial artery, that's because you know where the radial bone is, or the ulnar artery, and that can help you. The names are actually really helpful. So make sure you think about this and don't try to just memorize it as you go through. Then I will also ask you to know what part of the body, not what is the next vessel, but what part of the body does that ultimately feed if it's an artery. So for example, the internal carotid, where you might take your pulse, is going up to your brain, whereas the external carotid goes out to the superficial structures of your face. So that's what I mean by where is it going. If I say draining, then it's a vein. So analogous there would be the internal jugular is draining the brain, whereas the external jugular is draining the outer part of your head and facial structures. So that is, I give you lots of chance to practice these things, as you'll see in the study guide. And one of them are these things at the very end. And there are lots of ways to answer. Sometimes there's more than one answer to this, but we can go through this before the actual lab test as part of our review, but do try these on your own. And I believe we won't deal with that yet. Yeah, so we'll deal with this when we do the blood pressure lab on Thursday. So let me go away from this and share your list of blood vessels, which I believe I left here as well. Yes, I did. Okay, so now I'm going to share the screen again. Here we go. So this is actually, I'm going to go to the beginning of this PowerPoint. Let me get to the, let's see if it shows up reasonably. Okay. Can I do the, well, let me go to the very beginning of it. we talked about this. Is this going to, now it's not going to show up okay on this. It shows up okay in yours, but this is actually right out of your lab manual. I'm sorry, that's not going to help us because I thought it would show up all right. It looked okay when I just was in PowerPoint, but I guess a jumble happened when it went through Canvas. So these are pictures from your lab manual that I literally took out of your lab manual. And you'll see that not all of the vessels are ones that you'll need to know, but this is the whole body. So most of these you would, since they're some of the largest arteries of your body. Whereas something more detailed like that, I don't have you learn every single one of these guys. Now, generally, these pictures are pretty good. I think one of the struggles is that they only have arteries and then they only have veins. They don't have them overlapping in many places, so you can see how the artery and the vein run next to each other, have the same name. So the other part that I would like you to focus in on these kinds of pictures is there is an anastomosis that you need to know of, a particular and very important anastomosis, which is called the circle of Willis. So if you notice, going up towards the brain, this one's going in, and this one's going into the brain. You see them, they look kind of shadowy here. These are two large arteries. This one's called the vertebral for reasons I hope are obvious. These are the vertebrae. And this is your internal carotid in here. And these guys go up and they come to a common artery, a series of arteries called the circle of Willis. That is shown on this next slide. So here is a vertebral right here coming up, and there's a vertebral on this side. You have each side has its own vertebral. And then I know it's a little hard to see here, but right here and over here underneath this optic nerve is the internal carotid. So if blood flows up the vertebral into the basilar, it ends up flowing into the exact same sort of area that the internal carotid brings blood to. So this area right here is called the circle of Willis, where blood coming up from the heart through the vertebrals and then the basilar end up into what are called these communicating arteries. This is the posterior communicating, and this is the anterior communicating. So you can see they form kind of a circle, where the internal carotid can pour blood into the anterior, and for that matter, it could go into the posterior communicating. Now, when it would is if something happened to one of these vertebrals. So if you have arteriosclerosis, say, or you have an accident and the vertebral on this side no longer can contribute blood, then blood that would normally come up the internal carotid and go off into the anterior cerebral and other arteries could also flow through this communicating to the posterior cerebral and do the posterior part of the brain. So it is very common for the internal carotids as you get older to become occluded with arteriosclerosis. And people walk around and function. My dad had this happen twice. Fairly normally. I think the only thing he noticed is that math was a little harder to do. So, clearly you're not serving the brain as much as you'd like with blood, but because of this circle of Willis anastomosis, you survive it. And then they went in and, used a balloon to open up his artery, and it went back to normal. But it's fairly common to have those occluded and therefore give you the... So there is a list of the ones you're going to need to know from the circle of Willis. I pretty much went through them. The vertebral, basilar, you don't need to know these little branches off of it. The posterior communicatings here, the posterior cerebral and internal carotid, which you can barely see on this picture, and anterior communicating. And then this one's a little bit weird, because it actually doesn't go straight forward. It actually bends and goes into the fissure here. And that's just how they're trying to represent it. It's going to dive right into the fissure to do the anterior part of the brain. You can actually kind of see it diving in here. So that's the anterior cerebral. Now for the abdominal ones here, the abdominal arteries, there's a little trick slide I'll show you. I think I showed it briefly before to help you that I got out of a study guide, and then I used my little art program to imitate. So this looks a little bit like a man with his legs. Well, his belly button has an umbilicus on it, so maybe it's a baby. And then he's got two little things that would be where the nipples are, his nose, his arms, and eyes. And so they came up with this little study guide of the abominable abdominal man. And I put it over here to help you remember. So once again, you don't need to know many of these, like these guys up here, the inferior pharynx. I didn't even ask you to do those. Just the big ones that are on here. So even though I put the inferior pharynx, they're not on our list. The only reason I included them is they're the little eyeballs on the man. If something is called a trunk, that's because it's going to very quickly branch into two largish arteries. So you know of the pulmonary trunk around the heart that really quickly becomes the pulmonary arteries. Here you have the celiac trunk very quickly becoming the splenic. Now the splenic goes to not just the spleen, but the stomach. So it's called the splenic, but it goes and becomes the gastric pretty quickly. And then over here, hepatic, that means liver. So this one's going to bring oxygenated blood to the liver. Renals should be self-explanatory, that's kidneys. Gonadals, hopefully that's self-explanatory. And then you have these mesenterics. Now they're called that because they go to all of the intestines. And the way they get there is there's connective tissue that's kind of suspending your, it's actually serosa that's suspending your intestines, and now that's the mesentery. So for the inferior and superior mesenteric, they go, because of all the anastomoses, they go to all of the intestines, both of them basically. If you want an answer for who these guys feed, it would be intestines. So small and large together. Now let's see if this one looks better. I don't know why the artery one looks so bad, but these are in your lab book, so they could help you with the answers to who feeds what, because they are showing that. Let me give you a warning. No answer is going to be aorta or inferior or superior vena cava. So I'm not going to ask who the inferior vena cava drains, because it's most of the body, right? Or I'm not going to say who does the aorta feed, because it's all of the body except the lungs. So yeah, so we're mostly focused on these guys that are away from the heart. Okay, so be careful of your answers. If I were to pick, say, this common iliac, now when something has something like common in its name, that's because it also is going to branch pretty quickly. So the common carotid, the common iliac quickly become like internal and external carotid, internal, external iliac. If I pick the common carotid, then you need to be clear how it differs from, say, the external common iliac, how it differs from the external iliac. So for example, the common iliac would be doing your gluteal area, your pelvic area, and your lower limbs. Versus if I said external iliac, then you could say the entire lower limb. Does that make that clear? If you find these figures useful, then go for it. There are one set of vein names that are kind of funny called the azygous system. The word azygous means unpaired, and I don't have a picture of it here, although we could look at at it. Well, maybe I do in this. Does it show it? No, it doesn't show it. That just means the right and the left side look different from one another. So on the right side, you're going to have the zygous veins draining the posterior thorax. And on the left side, you're going to have the accessory hemiazygous and hemiazygous system draining the left side. So their names are a little different on the left and the right. So that's how it gets, and they look a little different unpaired. Oh, I do have a picture. Here it is. So here's the azygous, and then here's the hemiazygous and accessory hemiazygous up here. These guys drain blood into here, into the azygous. So it would be fine to say the azygous drains blood from the right posterior thorax and the hemiazygous accessory from the left. It's true that the left drains into the right, but in general, the goal of the zygous over here is to do the right side of the chest. Okay, and here you can see there are extra ones. Like you do have, you have a radial vein and you have an ulnar vein, but then you also have a basilic vein. You have a brachial, which, okay, that makes sense. But you also have a cephalic. There's just more veins than there are arteries. Okay, so this is your list. It is abbreviated from what's on those figures in the book. And I found this in the textbook. Again, it's kind of like this diagram, but done in more of a, this is connected to this way, so that it's not that you need to know everything on here. The list you need to know is over here. But I thought, like here, it shows the circle of Willis nicely. Shows the vertebrals coming up to it and the internal carotids coming up to it. Whoops, internal carotids. Where are the internal carotids labeled? Oh, they are labeled there. Okay. So, and the communicatings. So, if these are helpful, they're there. I just put them in there. You don't have to do anything with them. But what I thought, besides this being the list, you could do over here is you could write the answer to who this artery feeds. It would be fine for the vertebral to say the whole brain, because through the circle of Willis, it can do the whole brain. That's good enough. You don't have to get more specific. So that would mean internal carotid would have the same answer because of the circle of Willis, the whole brain. So if you want, I don't collect this, it's just to help you study. Now, these numbers in parentheses are not useful for you. Unfortunately, if we were in person and we had the wire model, which you'll see in pictures in a minute, these numbers correspond. So that way you had a key to the wire model. The wire model has a lot more vessels than we're studying, and you could just look for that number and you would have been able to study your blood vessels. But in my photographs, you can't see the numbers very, very well, so they won't help you. So you can just ignore these numbers or letters that are showing up after this in the list. Okay, so that's a bunch of tools. Let me stop sharing this. And, okay, and go back to, I guess I can't.
Professor?
Yeah.
Will you be posting the presentation that you just shared with us? Or is it already posted somewhere?
You don't allow me.
I see. Okay.
Yeah, but mean that PowerPoint, you want that?
I think some of the figures I found under your learning aids for vessels, but I was just wondering if some of the other figures were in there.
I think they're all in there, or they're in the study guide. That's just an old study guide I had. And I just thought it's a whole PowerPoint slide I had to introduce the lab. But I don't think there's anything that you don't have already somewhere. I just used it because it had a mixture of the lab manual pictures and the study guide pictures.
Okay, thank you.
Let me see. There's a bunch of chats. Let's see what's in it. What is the PPT? Or where is the PowerPoint? Is that what
Yeah, I
that
think we were
says?
just trying to look for that, basically, yes.
Yeah, no, it was something I had from the past. I mean, if you really want it, I don't mind putting it up there. So I can go ahead and put that in files, if you'd like. Yeah, so I've kind of moved them around over the years, so they're all there. They're either in your lab manual or they're in the useful, which is what I was going to show you next. And then Vincent said, can we go over the ECG assignment? I believe that's due Thursday, right? Why don't we go over that Thursday? Because you don't have to turn it until 5. And because the blood pressure lab, I won't have so much, we'll have some introduction, but I'd like to give you guys time to work on some of these pictures and models today.
I think you have it listed as due today, no?
Is it
Yeah, I
due
see
today?
it is due today.
Yeah, it's due
Oh,
today.
well, all right, then we'll go over it quickly if I can. Okay, so, well, let's quickly share I was going to go back to this thing, I guess. Maybe we shouldn't do that. I could use this time. Let me drag this up. I was just going to show you, this is in words, what we just did before and this is the kind of thing that could show up on the test as well. I might just use words and not pictures. Okay, so you want to back up to the ECG. You want to actually do the handout? Is that what you wanted to do? Let me get back to the chat so I can see it. Is that what you were talking about, Vincent?
Yeah, I think he's talking
Yeah.
about the table.
Okay, so I think if I go back out of here, here is the ECG assignment. Are you still sharing? Are you still seeing?
Yes, we can see.
Okay, so the P wave, just anybody yell out, what's the electrical event that's causing the P wave?
Atrial depolarization.
Great. And if there are no P waves evident, although QST waves are present, then what's not functioning? What do you have to
The ventricles aren't working.
What'd you say, Adrian?
Oh, sorry, the atrium aren't working. There's no P waves.
So there's some.
Did you say AV or?
It's through the SA node, the sinoatrial.
Right.
That's
Okay,
not
so
firing
that
properly.
is the reason. The SA node isn't firing. The atria are probably in atrial fibrillation, meaning that they're not, contracting together, but the AV node is taken over. So in that person, not only will their waveforms look different, they'll be missing the P wave, but what would happen to their heart rate? Faster or slower?
Slower.
Slower. So they would probably have bradycardia. And what would happen to their end diastolic volume? Increase, decrease. Remember, the atria are not contracting together.
Decrease.
Decrease. So this person is alive. They may not want to run a marathon or run anywhere, but their ventricles are being driven by their AV node. Okay, how about the QRS wave? What electrical event is causing that?
Ventricular depolarization and repolarization.
I can barely hear you. Was that you, Jessica?
No, that was someone else.
Okay. Who's got an answer to what is happening electrically in the heart for the QRS?
It would be ventricular depolarization beginning at the apex and atrial repolarization. I think that's what they were saying.
Oh, okay. Yeah, and in fact, what's underlying this you can't see is also the repolarization of the atria. So this is ventricular depolarization. And if there are fewer QRS than P waves, but they're at regular intervals, Now, this is where that idea of a blockade came up, and in the videos, they distinguish two types. One that you have any prolonged interval here, and that's a type 1, and then type 2 blockade. The AV node is just not being stimulated by the SA node reliably. So you get the P wave because the atria depolarizing, but occasionally, or it could be often, the AV node is not firing, and then you have an AV blockade, often because there's something wrong with the ability to bring the AV node autorhythmic to threshold efficiently. So what about this interval? Can anybody give me a time frame that you would fill out in here for the PR interval?
At 0.12 to 0.2
0.10
seconds.
to point.
I think what I have is 0.12 to 0.2 seconds.
Right. Okay. So 0.12 to 0.2 seconds. So that's this interval. And during that interval, you're conducting current from the SA node to the AV node, and you're getting the AV node to fire because that ends at the beginning of the Q or during the R, since we're not, this was the one that Kevin did the PQ. But that's fine if you want to say that's what's occurring there. And if it's prolonged, we talked about that being a first degree AV block where it's just taking longer for that amount of depolarization to get the AV node to fire. The QRS interval, anybody have a time frame on that one?
Is it 0.06 to 0.12 seconds?
Yeah, I've seen 0.1, but that's okay. 0.06 to 0.1, or what did I put in my handout? Let's see if I think I put, yeah, 0.06 to 0.1, but that's fine if you found that somewhere in the videos. Okay, if that's prolonged, now I don't know that this was covered in the videos, but it's often due to a bundle branch problem. Because after all, this is depolarization of all of the ventricle together. If it takes longer to get them all together, then it's because traffic of action potentials through autorhythmic has been slowed or disrupted, and often that means one of the bundle branches isn't working, so you're counting on some of that depolarization to be spreading just from cardiac contractile to cardiac contractile. So you've got potentially a bundle branch block here. Okay, what's happening from Q to the end of T, the beginning of Q to the end of T?
I have down ventricular systole from ventricular isovolumetric contraction to isovolumetric relaxation.
Great. Those are the cardiac cycle phases, which is good to integrate, but this table is actually asking about electrical events. So you're getting the depolarization in order to get, you're right, the isovolumetric contraction that is occurring because they've depolarized through repolarization. So the isovolumetric relaxation would occur then. So you are good at correlating that, but this particular box asks for the, what did I have over here? Normal range. Oh, the normal range is here. And what is a normal range for that? Anyone come up with some numbers?
I've got 0.31 to 0.41 seconds.
Great. So that's what I have too. And if it's prolonged, if that whole thing of depolarization, repolarization is longer than normal, is there anyone who came up with why that could happen?
I put that one of the issues could be that like electrolyte levels are low.
Particularly potassium is affecting it. So that's where the T wave gets prolonged because there's a potassium imbalance. And after all, repolarization depends on that potassium. There's also some genetic underlying causes that people may have in their gated channels. Okay, so that's all prolonged. I didn't shorten any of these, but if you cause sympathetic activation, remember you're allowing calcium to come into the cardiac contractile fibers, and that is contributing to stronger contraction, but it is a positive ion. So it tends to shorten these intervals. So that was the idea of what he was doing when he was exercising, and we'll get to that in a second. Okay, I hope you can define all of these. The only one we didn't do is asystole in the past. What does it mean to be asystolic? A means
No electric activity.
it means there's no rhythm. If asystole, there's no obvious rhythm to what you're seeing in the depolarization. Flat line could be an asystole where you're no ECG. You're without a systole. Okay. So then you put in his data, and hopefully you have all that. And I'm hoping that what you saw, and I believe I wrote his data down, is that you get a shortening of his, when he's exercising, of some of these intervals. Now, I didn't ask about TP. That's the time from the end of the T wave to the beginning of the next P wave, sometimes called the quiescent period. And there isn't any normal range because it really depends on heart rate, your resting heart rate. If that's the time between. So why does exercise shorten heart rate? Well, think about the calcium bringing positive charges in and what that's going to do to bringing the cell to threshold faster. So, or if it's an autorhythmic, then sodium is being brought in, so it doesn't even make it to -60 before it starts going and depolarizing. And the steepness of the pacemaker gets steeper, so you end up with shorter intervals with the autorhythmic too. Are we good on this now? Okay, so turn this in by 5 tonight. I've been noticing many people are turning in late, and I've just forgiven that. But since this one was done, hopefully done, before you worked on it before, then I would like this by 5 o'clock tonight, if at all possible. Lateness, I take 10% off, so it'd be like half a point or something, but still, I haven't been up to now. Okay, so if you would like to be into a study group, I will put you into that study group. You have about an hour, is that right? Not quite an hour to work. And what I really encourage you to do is look at the pictures. I guess that's what I was going to show you of our models, but I'm sure you can find them. I could go back. Where you're looking is under this gross. Oops, am I still sharing? I don't know if I'm still sharing or not.
No, I don't think so.
No, okay, let me share again. Okay, this one, share. You're looking in the, here's that list of vessels to know. That's that handout I showed you. And then you have the vessel model photos. And there's a lot of them. See? They're often redundant. They're just, sometimes you're seeing them on a lower, this is the arm model that we studied for muscle. So, or this is the pelvic floor that we studied. In this case, it's an isolated set of abdominal muscles that came out of the older torso. And then there's a few these are things called relief models. I'll show you one of those. Where the vessels are, it was made to show you vessels. Be aware with this guy that this is showing you the anterior part, and this is trying to show you the posterior part. If the vessels like this end in dead ends, they're just somebody's imagination half the time. So most of these you would, you'd only have to know the big main ones that are going, like these guys here. So anyway, this is a useful model. I've got a picture with all of them together, and I also have a picture of just the bottom like this and just the top that it's a little bit bigger. Let me close this, but I got to move you guys to do that. Okay, so that's all useful. And then if we go back to this guy, there's some things in here that I briefly showed. This was something a student made a few years back. So this is generated by one of you. I only have it as a PDF, but it looked pretty good. So, it helped kind of show where they're arranged in the body. If you'd like that, that's there. It's, it's just another tool. And I helped you more than I was going to, but once I looked at the pictures and tried to do this myself, here are all those pictures again, right, that I just showed you. I suggest you don't go into this until you try to do it yourself. I labeled them for you. But I suggest you try yourself to identify it and then use this as a key. Okay? So lots of tools in there for your enjoyment. And lots to do. So let me stop sharing again and let you guys go. And it doesn't look like anybody has put into the chat that they would like to be in a breakout room, but I'll be here for the next 45 minutes or whatever you have, 50 minutes. So just let me know if you need some help. Or you decide you do want to go and look at these pictures together, then you're welcome to do that. Okay. All right, so turn in the ECG handout by 5. And then on Thursday, we're doing blood pressure, but Kevin never sent me any of our power lab. I think it was probably hard for him to do that lab by himself as a demo. So what we're going to be doing is looking together at one in the mastering that's a little different. It goes through it. We won't have any result table to fill out, but we'll go through a lot of the concepts. So I'll probably be walking you through a lot of the blood pressure stuff in the lab that you would normally be studying by doing. Okay, all right, just give me a shout if you need anything. I'll be here, although I might be on another screen. All right, we'll see you Thursday otherwise.